All right. Hello, everyone. Thank you for joining us for the third annual Friends of Biology gathering. My name is Daniel Griffin, and I am the Development Officer in the Biology Department. We prepared a wonderful lineup for you today, featuring remarks from the Dean of the School of Science, Nergis Mavivala, followed by an update from Biology Department Head Alan Grossman. We will then have presentations from 3 current graduate students who will be sharing highlights from the research taking place in each of their labs. Alan will facilitate a brief Q&A session following each of the students' presentations, and upon conclusion of today's prepared program, we invite any of you who would like to continue the discussion to join Alan for a virtual reception, an opportunity to connect with one another following the presentations. That link is currently available in the chat, or should be shortly if it isn't, and we will share it again at the conclusion of the program. A few housekeeping items of which to be aware. This portion of the program is being recorded, so anyone who could not join us live will be able to view this at their convenience, and we'll be sure to share the link to that video once it has been posted. Each of you can submit questions at any time through the Q&A function, which hopefully is readily visible to everyone. We'll be monitoring the questions as they come in, and we'll do our best to answer as many of them as we can throughout the session. You can always contact me, Alan, or anyone in the department following today's gathering via the biology homepage. Lastly, if you have any difficulty or experience any technical issues during the program, please email earmy, that's EARMY, at mit.edu for assistance. And you can also email me, as most of you should have my email at this point as well, and we'll do our best to try and help. In my role as the department's development officer, I'm very fortunate to work with a wide variety of supporters, partners, volunteers, and alums, many of whom are with us today. And I want to thank all of you for your ongoing commitment to our work, as we simply could not do it without you. So before I hand it off now to the Dean, I want to acknowledge the Alumni Association for their help coordinating today's event, as well as the Club of Boston in helping spread the word. We are thrilled to have so many of you here with us, and I hope that today's program provides you with a sense of the scope and breadth of the teaching, learning, and research that is taking place across the department. So with that, here are remarks from the Dean of the School of Science. Thank you very much.
Hello, everybody. It is my great pleasure to welcome you all to this gathering of the Friends of Biology. I'm Nergis Mavivala, and I am the Dean of the School of Science at MIT. And I have the pleasure of having the Biology Department be part of the School of Science. We are all, I'm sure, acutely aware that we're here meeting together in digital space rather than getting together with each other in physical space, and that there is a global pandemic that has really altered the way we live and work in the past year. I want to use this moment to ponder that the pandemic has highlighted for us some of the ways in which MIT as unique and MIT biology in particular has played a tremendous role in shaping how this pandemic played out. I should remind us all that some of the most important therapies and technologies behind the vaccines that will hopefully get us out of this pandemic were invented here at MIT 30, 40, 50 years ago. And indeed, it's a reminder to me when I think about the role of science that some of the discoveries of today, in fact, most of the technologies of tomorrow are predicated on the discoveries of today. So I couldn't be prouder of the research that's been going on in the school and in the biology department. As an educator at a university, I also must highlight the enormous role that the students play in everything we do, including thinking about how we deliver education and do research. Everybody has pulled together in a phenomenal way over this past year through all the disruptions in keeping our education and research missions going. If you haven't had a chance to check it out, the biology department spearheaded an online course on COVID and SARS-CoV-2, which has been a tremendous success, and certainly I've learned a lot from it as well. And so, I also want to take a moment to highlight that everything that we do here in the school, in the department, is enabled by the generosity, the partnership, and the engagement of the friends and supporters of our school and of the biology department. So I want to thank you and welcome you to this event. And I sure hope that next year we can be together in person and that I can meet you all. Have a wonderful time.
Thank you, Nergis. Thank you also, Daniel and Lizzie, for helping put this together. I do want to point out that Nergis is a physicist, and I think in this video, she was somehow playing with the time-space continuum. There was a little bit of a disconnect there. I think maybe the other possibility is that it was a Zoom artifact, but Nergis is a physicist. My name is Alan Grossman. I'm professor of biology and currently the head of the Department of Biology. I've been department head for about 6.5 years. I don't want to say how many years I've been on the faculty, but I did start in 1988. It's really, I'm thrilled to be here to talk today. I'm thrilled that so many of you have been able to join us. I can't see faces, but I do see a list of some of the participants, and it's really exciting to see so many friends and colleagues and many alums, former students who I know quite well. So what I'm going to talk about really is some of the stuff that's been going on in the past year, and just sort of my perspective as department head about how we're coping with the current pandemic and some of the really exciting things that I think are going on in the department. For me, it's just an honor and truly a pleasure to serve such an outstanding department. Our mission really is to continue to maintain and generate excellence in research, teaching, education, our mentoring, and service to internally and service worldwide. To me, one of our greatest strengths is the people, and it's just, again, a tremendous honor to work with such wonderful people, the students, the postdocs, the staff, the faculty. And it's also an honor really to have such dedicated supporters and volunteers and friends and alumni who really help to enable us to fulfill our research and educational and service missions. I think we're all part of a really special community. The department has about 60 faculty members. 3 of them currently active faculty are Nobel laureates. About half of the faculty members belong to the National Academy of Sciences. So it's a really outstanding group of scientists, but also an outstanding group of people. The graduate students number approximately 230. We take about 35 new students a year. There are about 190 to 200 undergraduate majors in the department. Most of them are doing research or will do research at some point. And we have 3 different flavors of biology majors. There's the traditional course 7. And for some of you, we now have course 5-7, a chemistry biology joint major, and we have now for many years a computer science and biology, a 6-7 joint major. And the 6-7 major is actually quite popular now with the emergence of machine learning and artificial intelligence, and combining that with biology. For me, and I think for many in the department, one of the most rewarding and important things that we do is training, is teaching people how to be scientists, teaching people how to do research, teaching people how to behave like responsible scientific citizens, and really just educating the next generations of biologists and scientists. These efforts for many, many years are obvious and visible across the country, across the world, actually, in industry and government and academia and medicine and public health and well beyond. And again, I take, as much as I enjoy the research that we do, I take great pride in the teaching and mentoring. That's really one of our top priorities. I think, as most of us are now aware, there's a pandemic going on right now. It hit pretty hard in March, about a year ago, March 2020. And that presented many challenges for the department, for the institute, and I think for all of us as individuals. What happened almost immediately was we were told to vacate campus. We weren't allowed to come to work. And we quickly had to pivot to teaching, continuing to teach our classes in an online setting. And we in the department and across MIT, we were able to offer all of our classes in the spring virtually, online, typically in this Zoom format. Not so much in a webinar, but in sort of more traditional Zoom format. This was a challenge, and we learned a lot from it. Some of it worked quite well, some of it maybe less well. And in the fall of 2020, we were still in this virtual reality. And based on what we learned in the spring, I think we greatly improved the student experience, and I think also our experience in being able to provide high quality education to a fairly large number of students who were learning remotely. Some of my colleagues have been significantly more creative than I in creating virtual backgrounds. Daniel, if you might have noticed, is either in San Francisco or likes the visual of that bridge. But somebody, Barbara Imperiali, who's teaching introductory biology right now, creates a different virtual background for each one of her classes. And they're usually famous places or buildings or really beautiful scenic settings. To my knowledge, no one has yet turned into a cat, but we have a little ways to go for the rest of the semester. The pandemic immediately in March significantly reduced the research enterprise. We were not allowed to come to campus. That changed a little bit as we moved along, and by the summer we were functioning at about 25% capacity, and then eventually to 50%. And now we're still functioning more than 50%, but less than 100%. So the research enterprise, I would say, is back up and rolling along, but not quite where we would be in normal times. I do want to mention the COVID course that Nergis talked about. In April, May sort of timeframe, it became obvious to me that there was a tremendous thirst and a desire and a real need for information about the pandemic, a need in our community. People who were not biologists had lots and lots of questions about what was going on. And there was a need nationwide for information about the disease, the virus, vaccines, diagnostics, possible treatments. And so I thought it would be a good idea for us to offer an online course that focused on sort of the basic biology of the pandemic, the virus, the disease, and sort of outlined what such a course might look like, and then talked to a few colleagues in the department, and everybody thought it was a good idea. And so I asked Rick Young and Facundo Batista to organize such a course. And they put together something that was successful beyond certainly my wildest dreams. The COVID-19, SARS-CoV-2, and the pandemic class. There were 14 lectures, 14 guest speakers, outstanding speakers, all luminaries in the field. And these were available in real time online for anybody who was interested. And the videos of all of these lectures are freely available and they're still available. If you're interested, just go to the biology department website. There was a special section for our students if they registered for credit. They got to interact directly with the speakers after each lecture, and there was an outstanding TA involved in this course who worked closely with the undergraduates who took it for credit. To date, we've had over 500,000 views of the 14 videos. I think the most popular has been the talk by Tony Fauci. So if you've not seen these, or if you're interested or want to see them again, I encourage you to visit our website, and those videos are freely available and still posted. The feedback from this course has been outstanding. People really loved it. They were reassured to have reliable information, accurate information. And recently we decided we're going to run this course again this coming fall. There is a tremendous amount of new information available about the vaccines and how effective they are, how they were tested, the various virus variants and how the vaccines are effective or not against those, potentially new treatments. So the fall course will have up-to-date information again, and we will also have a constellation of outstanding speakers. We've not organized the course yet. That'll be done over the summer as we have a clearer view of what will be the most interesting and most topical. The other thing I want to say about the pandemic is that MIT has really created a safe work environment. Anybody coming to campus regularly is tested for infection at least twice a week. And there's vigorous contact tracing. MIT is doing about 20,000 tests every 5 days. And the current positive rate is less than 0.1%. It's about 0.05%, which is significantly lower than the rate in the general population. So the campus is quite safe, and people again are wearing masks, practicing appropriate hygiene and care. There have been lots of opportunities presented by the pandemic, as terrible as it's been and difficult as it's been to get through. This virtual setting and us getting used to Zoom has enabled us to have speakers from all over the world come and save tremendous on travel costs and carbon footprint. For thesis defense seminars, have had typically 80 to 100 people with parents and siblings and relatives from all over the world, which would not be possible when we're in person. So I think when we're back in person, we will learn from what has happened and take the best things that Zoom and the pandemic have had to offer. And we will have thesis defense seminars in person, but we will probably live stream them so that we can include people from all over. There have been tremendous challenges personally to many in our community. And I'm super proud of the people in the department, especially staff who have been working incredibly long hours and hard to just make sure that we can keep doing what we're doing. And the toll on our students and postdocs and faculty has been significant, but we are getting through it. We are also doing a lot of things that we normally do, only we're doing them in this virtual setting. We are continuing to interview faculty candidates. We have open faculty positions. We're in the process of interviewing 17 candidates. We're going through our graduate admissions process the way we normally do, except we're doing it virtually. We had about 900 applications to our graduate program. Over 24 faculty members served on the admissions committee. They narrowed it down to about 100 students to interview, and we're about 3/4 of the way through those interviews, and we're aiming for a class of about 35 new students to start in September. Hopefully in September, we will be back teaching in person. That is certainly my hope. I did not really enjoy teaching by Zoom so much, but if we have to do it virtually again, we will. Looking ahead, there are many unknowns. I think most of the population will be vaccinated soon. Whatever these unknowns are, we will continue to work and strive to be excellent in all of our research, our teaching and education missions, and providing the best opportunities for all in our community. I'm cautiously optimistic about the future, and for me, the most important thing is to continue to support the people in our community. The people are our greatest strength, and it's the people who determine what we are and aren't able to do. And those, again, include our undergraduates, our graduate students, our postdocs, our staff who support almost everything that we do, and our really outstanding faculty. I'm grateful to all of you for participating in this, for your long-term partnership and helping to make all this possible. As Daniel said, I'll be available for more informal conversation in a Zoom setting, still virtual, but a little bit less formal than this webinar, and happy to meet with people there and take questions and have discussion. Right now, we're going to hear from 3 student speakers. I want to remind people that there will be time for questions and answers after each talk. And if you have questions to pose, please use the Q&A function. I will read the questions for the speakers to ask. And I apologize ahead of time if we're not able to get to everybody's questions. We will do as many as we can while trying to maintain some semblance of being on time. So it is truly my pleasure to introduce our first student speaker. That is going to be Grace Johnson. Grace is a graduate student in Jean-Wei Lee's lab in building 68. Grace, it's all yours.
Great. All right, so I'm really excited to talk to you guys today a bit about the research that I've been doing in Jean-Wei Lee's lab that I think really exemplifies the importance of studying basic biology in diverse species. And I'm going to focus on bacteria today. Bacteria are an incredibly diverse domain of life. They can live in a wide variety of environments, from the guts of mammals to oceans to soil. They can live synergistically with their hosts or cause severe disease, and they can really perform a wide range of metabolic functions, such as photosynthetic bacteria or bacteria that ferment yogurt, just to name a few examples. However, despite this really incredible diversity, a lot of how we think about the most fundamental aspects of bacterial biology has been informed by seminal work done in a single model bacterial organism, E. coli. And I'm going to focus on one of these fundamental principles today that's really been the focus of my research, that of transcription-translation coupling, which has been largely characterized in E. coli and other related bacterial species, and has really formed the basis of how we think about regulation across the bacterial domain. But what my work has shown is that this fundamental principle is actually not as universal as we once thought. And this has really important implications for how we understand regulation across bacteria. So what is transcription-translation coupling, and why is it so important for how we understand bacterial biology? So coupling involves, at its most basic level, 2 molecular machines, the RNA polymerase and the ribosome, and how fast they move within the cell. So these 2 machines are involved in a process that's central to all of life, from bacteria to humans, the process through which the information encoded within DNA genomes is used to make proteins that carry out most of the cellular functions. And this process occurs in 2 main steps. In the first step, the RNA polymerase reads the genes encoded in the DNA and transcribes them into RNA copies that are then read by the ribosome and translated into proteins. And these steps of transcription and translation that allow information to flow from DNA to RNA to protein are really so core to biology that this process together is known as the central dogma of biology. And so I've shown these 2 steps of transcription and translation as happening one after another. But actually in bacteria, they can happen at the same time. So what this means is that as the RNA polymerase is reading the DNA to make the RNA, the ribosome can begin to read that RNA and make the protein at the same time. And this is what's known as transcription-translation coupling. And one of the really important features of coupling that's been characterized in E. coli is that these 2 machines move at the same speed along their respective tracks. And this allows the ribosome to keep up with the RNA polymerase throughout this process and really maintain a close proximity to the RNA polymerase. And this close proximity allows these two machines to influence one another. And this forms the basis of a lot of the regulatory mechanisms in E. coli, or the way that E. coli senses signals from its environment and turns those into changes in which proteins it's making. And so the speeds of these two machines, and therefore this principle of coupling, has really formed the basis of how we think about gene regulation across bacteria. But whether or not coupling exists outside of E. coli has never been explicitly tested. So part of my research was to measure the speeds of the RNA polymerase and the ribosome in a distantly related bacteria, that of Bacillus subtilis. And what I found was quite surprising and interesting. So in this bacteria, the RNA polymerase actually moves a lot faster than the ribosome. And this means that the ribosome can't keep up with the RNA polymerase like it does in E. coli. And we have termed this form of transcription runaway transcription because the RNA polymerase runs away from the ribosome. And because of these differences in speeds, there's actually quite a significant distance between the RNA polymerase and the ribosome throughout this process of transcription and translation. And this means that these two machines can't influence each other in the way that they do in E. coli. And this actually helps explain why the ways that Bacillus subtilis regulates its gene expression are quite different than the way that E. coli does it. And so this work is really exciting because we've been able to use this finding of this very basic difference between E. coli and Bacillus subtilis, this difference in speed between these two machines, and use that to help us explain how different regulatory mechanisms have evolved between different bacteria. So clues from genomic sequences of various bacterial species have indicated that Bacillus subtilis is not just some weird outlier, but rather that some bacteria use coupled transcription like E. coli, and others use runaway transcription like Bacillus subtilis. And so this really has changed how we understand the central dogma in bacteria, because coupled transcription is not universal, but rather one of two key ways that bacteria can express their genes. And I think this work really highlights why, even at the most basic level, it's important to study diverse organisms, because they can give us really interesting, or teach us something really interesting about how biology works and how it works differently in different systems. And I also think that this work is really interesting because it's been able to show how a very basic principle, the speeds of these two machines in the cell, are really able to help us explain some of the evolution of the diverse mechanisms that we see across bacteria. And with that, I'd love to take any questions that anyone might have.
I'm going to applaud for everybody. Thank you, Grace.
Thank you, Alan.
that was great. I must say I've heard some of this before, and it's still, I love it. So Mark Snyder is asking, are there advantages to one mechanism versus the other?
Yeah, I think that's a really interesting question. I don't have a great answer as to what the advantages of one versus the other might be. And I think that one of the things that that's an active area of research that I think could maybe give us some clues to that is really how runaway transcription has shaped regulatory mechanisms within Bacillus subtilis. So obviously Bacillus subtilis is a well-studied bacterial species, and there are a lot of mechanisms that are quite different from the mechanisms in E. coli. But with this finding that runaway transcription occurs in Bacillus subtilis, trying to better connect this underlying phenomenon to how that has specifically shaped the regulatory mechanisms, I think will continue to shed light on the pros and cons of these two mechanisms.
Mark Samuels asks, says, very interesting stuff. Can you give a brief example of how the two mechanisms lead to different types of regulation of expression? And sort of, I'm going to couple that with a question from, I don't know if it's Jan or Jan Urenka. Sorry if I mispronounce names. In runaway coupled transcription, is there less communication between RNA and ribosomes than when coupled? And I think, again, part of that is what are the regulatory consequences?
Yeah, so I'll give there are a few regulatory consequences that we've looked into of these two forms of transcription. So I'm going to talk about, I'll just talk about one. So one of the ways in coupled transcription that the ribosome can really influence the RNA polymerase is by blocking specific signals in the RNA that can cause the RNA polymerase to stop transcribing. So obviously these signals are important when you get to the end of the gene. You don't want to just keep transcribing forever. You want to eventually stop transcription when you get to the end of the gene. But also sometimes the cell wants to stop transcription of a specific gene in response to a specific type of stimuli. And the ability of this ribosome to either block or promote this stopping of transcription based on how close it is to the RNA polymerase is really important for, is a really important mechanism through which the cells can sense their environment and translate that into actual gene expression changes. And so in Bacillus subtilis, it's actually really interesting because they don't have the RNA polymerase to modulate the termination of transcription in the same way that E. coli does. They have to use other means through which to modulate transcription termination in response to signals. So Bacillus subtilis can sometimes use other proteins to change when transcription is going to be terminated, or even sometimes use different structures of the RNA to modulate when transcription will stop or not. And so this is just one example of how kind of the differences in the type of transcription necessitate different mechanisms between these two bacteria.
I'm going to couple a couple of other questions again. So Guy Plunkett asks about phylogenetic patterns in the distribution between the two different mechanisms. And Tom Pollard asks, if you break the mechanism in E. coli so that it's not coupled, you know, what are the consequences of that? So is the coupling an optimization, or you know, what happens if it's not coupled?
Yeah. So I'll answer the first question about phylogenetic distribution. We see very widespread signatures of this runaway transcription in this bacterial clade that Bacillus subtilis is found in, firmicutes. But it's not just confined to closely related species in Bacillus. Other distinct clades of bacteria, including some more distantly related bacteria, such as Campylobacter, also have signatures of runaway transcription. A lot of these, I think we don't have a good answer as to, you know, I've described how runaway transcription and coupled transcription can shape the gene regulatory mechanisms of different bacteria, but we don't really have a great answer yet of that in the reverse, how maybe the biology or the environment of these bacteria have shaped whether they have coupled and runaway transcription. And I think that that's going to be something really interesting to explore going forward. And then as to what happens when you break this coupling in E. coli, I think that this is a really interesting mechanism and another example of how Bacillus subtilis and E. coli are quite different. So in E. coli, when you actually break this coupling, for example, an RNA that's normally translated, you add a stop codon, so a signal that prevents it from being translated, you actually get premature termination of transcription of RNAs that aren't being translated. And this actually acts as a really important quality control mechanism in E. coli, because it allows you to stop the transcription of any gene that's transcribed without this trailing ribosome, and therefore really prevent transcription of genes that aren't going to form a functional protein. So kind of aberrant transcripts that you wouldn't want to have around anyway. It's really interesting to think about this mechanism as well. Obviously, this type of mechanism does not work in Bacillus subtilis because the two are always uncoupled. So that doesn't really tell you anything about the fidelity of the transcript. It's another interesting aspect to think about what other stress response mechanisms might Bacillus subtilis have in place of that.
Okay, I think we need to move on. Thank you, Grace. I apologize to the dozen or so people who've posted questions that I can't get to, but lots of interest. Thank you, Grace.
Thank you.
Our next speaker is Bridget Begg, who is a graduate student in Chris Burgess' lab. And Bridget will tell us about some of her research. Bridget, it's all
Thanks,
yours.
Alan. So hi, everyone. Thanks for coming today. And thanks, Grace, for that awesome talk. I'm in Chris Burgess' lab, and one of the things that we really love to study, and I really love to study, is alternative splicing. I actually fell in love with it in college and I'm lucky enough to do it for my PhD. One of the exciting things about alternative splicing is that alternative RNA splicing really diversifies the output of the genome. It does this by having an alternative exon that can be regulated by untranslated sequences and upstream and downstream intron. And whether or not you include this alternative exon can produce two different transcript isoforms, one inclusion and one exclusion isoform, and these can result in different proteins that are produced from the same unit of the gene. And this really underlies a lot of the complexity in mammalian genomes. So the way that these introns are actually exert their regulation are mediated through RNA binding proteins, which are crucial regulators of gene expression really at all parts of the central dogma between transcription and translation. And they really do this by binding directly to the RNA and exerting their effects that way. And that's what we like to study in the Burgess lab. One family of RNA binding proteins that we really enjoy studying are the RB-Fox family of binding proteins, and that's because they're highly specific. They bind the sequences GCAUG and GCACG with high affinity, and also individual mutations in them are associated with autism, epilepsy, and cardiac pathologies. And although they're difficult to study separately because they're paralogs and they're family members that are very similar to each other, RB-Fox triple knockout cells fail to progress to mature neurons in neuronal differentiation experiments. So we know that they're absolutely essential for neuronal development, and particularly late neuronal development. In fact, all three RB-Fox proteins really peak in expression in a characteristic way in neuronal differentiation. But despite knowing exactly what these proteins bind and how important they are, GCAUG actually only explains about 40 to 50% of RB-Fox binding in any given cell type. And so what we wanted to know in the Burgess lab was, is GCAUG the whole story? And I'm going to bury the lead and say it's absolutely not the whole story. You can read about this in our paper from last year, but I wanted to just go over a couple of the conclusions from that paper and then walk you through sort of 1.5 important analyses really illustrating our findings. So in our paper, we show that RB-Fox binds a set of moderate affinity motifs in vitro and in vivo. We call these secondary motifs, and they activate splicing in an RB-Fox concentration dependent manner. So that depends on the expression of the protein in the nucleus. This property allows variation in RB-Fox and protein expression to have a two-wave model of splicing that's mediated by these motifs. So you're really able to activate different regimes of alternative splicing. And we think that this paradigm is broadly extensible to RBPs. I'm going to show you a little bit of data that really, I think, illustrates why this two-wave model could be really important for our understanding of gene regulation. We looked at a neuronal differentiation data set where we were differentiating mouse embryonic stem cells to glutamatergic neurons. And what you can see here is that over the stages, there's a really characteristic peak in RB-Fox expression. So I'm showing you on the y-axis. And if we zero in just on this particular peak, that's very characteristic, again, of neuronal development, you can see that from the radioglia stage to developmental stage 3, there's a huge amount of RB-Fox family expression. This is very interesting because this stage actually is a very diverse stage in neuronal differentiation, where the neurons go from being fate specified to outgrowing their neurites. They enter a dynamic state of trimming and pruning of the dendrites before they really have many characteristics of a mature neuron. So what we can do is study the secondary motifs that are influencing the alternative splicing across these time points. And what I'm showing you here is the association between the number of motifs, so 0, 1, or more than 1, with the splicing activity of RB-Fox, which for those in the know can be approximated with delta psi. So what you see here is what we would expect for a protein that has a strong influence on alternative splicing at this particular phase. And you can see the highly significant association between primary motifs, GCAUG and GCACG, and RB-Fox activity. This is between the first interval from the radioglia stage to developmental stage 1. If we look at the association between the number of secondary motifs and RB-Fox activity at this interval, you see that there is no significant association between motifs and RB-Fox activity. However, if we look at the next stage, which is developmental stage 1 to developmental stage 3, you can really see that secondary motifs begin to be engaged and are as significantly associated as primary motifs, which is really hammered home for us that at this point, it's really the increase in RB-Fox expression that allows a whole second cohort of alternative splicing changes to be occurring in neuronal development. We dug in just a little bit more to those events where primary and secondary motifs were both significant. So this is from stages developmental stage 1 to developmental stage 3. We looked at primary motif mediated events and looked at their associated gene ontology terms and found that they were heavily enriched in membrane and cytoskeleton terms, which is reminiscent of this fate specified and neurite outgrowth phase. However, when you look at secondary motif mediated events, what you can see in this interval, there's really more of an association with dendrite development and cell signaling, which is really a property of later stage neurons. And so what you can see here, and I hope what I've illustrated, is that these two waves allow the expression of one single protein to really dynamically regulate two different sets of exons, despite being able to bind the exact same types of motifs. So with that, I'd like to thank my lab and my co-authors, and I'm happy to take any questions.
Thank you, Bridget, on behalf of the audience. I'm curious, maybe you said this, what's involved in the specificity in determining the proportions of the different splicing forms?
In terms of the specificity of an RNA binding protein for a particular sequence, these are actually the primary and secondary motifs have overlapping shared different bases that really are the most important for the specificity and have the highest affinity. And then there are different bases that have sort of fewer hydrogen bonds that can tend to be a little bit more flexible. Interestingly enough, we also see this sort of bear out in conservation. If you look at sort of all of the GCAUG motifs in the genome, the internal bases are actually much more flexible in the conservation signal than the bases on the end, the two Gs on the end.
And in the course of neuronal development or neuronal maintenance, you know, what happens if there's a sort of unbalanced amount of the alternative splicing?
That's a really interesting question. You know, for RB-Fox, it's very difficult to study what as sort of in sort of a broad sense, what the different splicing events can do sort of in aggregate. However, we know there are many individual events that are extremely important that can be pointed to for RB-Fox. For example, there are different sodium channel subunits that are regulated by RB-Fox that can be actually swapped in early and late development, or that can be introduced to sort of diversify what type of chemical signaling neurons can participate in.
Reynold Verrett asks, comparing immune and nervous systems, the immune systems developed an independent approach to alternative splicing for diverse receptors. Is this a convergent property developed in neuronal cells that also randomize structures of pertinent proteins? So sort of a comparison between immune cells and neuronal cells and the strategies for alternative splicing.
That's a great question. I'm not terribly knowledgeable about immune cells, but what I can say is that these RB-Fox family proteins, there are 3 of them. 2 of them are actually exclusively expressed in the central nervous system, and one of them is a bit more broadly expressed. And so actually the additive expression of these proteins, because they bind the same thing, can really be influenced by their co-expression. So basically looking at what RBPs are expressed in certain cells can let us know how high their concentration is going to be, and can sort of let us know whether or not these secondary motifs are going to be relevant or not for that particular cell line.
And has again, this is Reynold, is the alternative splicing seen in the immune system, is that known whether or not it occurs in somatic cells, somatic tissues? And I'm going to follow up with sort of related, is how much of the alternative splicing is determined by cell type specific factors?
That's a great question. There actually is much of the alternative splicing is determined by cell type specific factors. So it's actually more associated, actually, if you study sort of the association among organs versus among species, you actually find a stronger association among organs of different species than differentiating between species themselves.
Okay, thank you, Bridget. I think we will move on. Our last student speaker is Charlie Shih. Charlie is a graduate student in Dave Bartel's lab. Charlie, all yours.
Thank you, Alan. Can you all see my screen? All right, so as you've heard from Grace and Bridget, the central dogma is this universal scheme by which genes encode the production of the molecules that together constitute life. And here I'm showing you a simplified view of this process in eukaryotes, where proteins are produced from mRNAs, which are in turn produced from DNA. But what's not shown here is another important process, and that is the regulated destruction of these molecules, which is important for controlling their levels and also for responding quickly to stimuli and other changes in the environment. And so today I'm going to tell you a brief story about this destruction in sort of two parts. And I'm showing here with these red arrows that proteins and mRNAs are constantly being destroyed by these distinct pathways. And in particular, cells express a class of tiny RNAs called microRNAs, miRNA here, which together target most mRNAs for destruction. And this destructive process is called microRNA-directed target degradation, and here's how it works. So this gray line is an mRNA that's being targeted via base pairing by a microRNA that's shown in red. And this microRNA works with a protein partner called Argonaute, or AGO, which recruits enzymes that eat away at this mRNA. And that's how the mRNA is destroyed. But importantly, the microRNA is protected by Argonaute and persists for many rounds of this mRNA destruction. And this whole process is quite well understood and is basically how most microRNAs work most of the time. But people have noticed over the years that sometimes certain microRNAs themselves are rapidly destroyed. But how this happens was not very well understood at all. And in fact, when you measure how long microRNA molecules tend to live before being destroyed themselves, most of them are indeed around for many hours. And you can see here in these distributions showing the lifetimes of every microRNA in mouse cells on the left and fruit fly cells on the right. And in both cases, the median lifetime is well over 10 hours, as you can see. But there's also this notable minority that lives for only a handful of hours, implying that they're being targeted for destruction by some mechanism. And my work has been really focused on figuring out what this mechanism is. And I'll just summarize here what I and my collaborators have learned over the past few years. So what we found is that when a microRNA pairs in a very particular manner to certain targets, here now these targets are shown kind of in cyan, the shape of the Argonaute protein actually changes. And this change is recognized by an enzyme complex in yellow here called the zesomate ubiquitin ligase complex. And this enzyme complex puts a mark on that Argonaute protein. And this mark is called polyubiquitin, and its effect is to target that Argonaute for destruction by the cell. And after that Argonaute protein is destroyed, the microRNA is left sort of naked and unprotected, and then it gets itself degraded quickly by various enzymes in the cell. And so to sum it up, in contrast to the canonical microRNA pathway, which destroys mRNA targets, we found that some targets can actually turn the tables on the microRNAs using this newly characterized pathway called target-directed microRNA degradation, thereby inverting the regulatory logic. And I just want to quickly show you my favorite result from our study, where every dot here is a microRNA that's expressed in either these mouse or fruit fly cells. And the x-axis on each plot shows how strongly each microRNA is affected by this pathway that we characterize. And the y-axis shows the lifetime of that microRNA. And what we see is a strong relationship between these two parameters in both mouse and fruit fly cells, meaning that the pathway really does explain why those microRNAs I showed you earlier have such short lifetimes. And we think this pathway is quite important in animal biology, especially in development. And some of my ongoing work is focused on answering the question of what happens in animal when this pathway does not function properly. And so with that, I'd like to thank you for your attention as well as your support, and I'd be happy to take any questions you might have.
Thank you, Charlie. One question from, I don't see anybody's name, but I see an email, which I will not attempt to read. Can cancer cells also be destroyed by a similar mechanism? So I think, can one target different RNAs in cancer cells to impinge on their ability to make tumors?
Yeah, that's a very interesting question. I didn't show this data just for lack of time, but we also profiled whether this process is happening in human cancer cells. And the answer is yes. And actually, the identities of those microRNAs that are being quickly degraded in cancer cells are largely the same as those in mouse cells. So actually these targets, first of all, the phenomenon, this process is conserved all the way through to the last common ancestor of bilaterian animals. So we see this process in worms as well as in flies and of course humans and mice. And in addition to the process being conserved, these microRNAs themselves are largely similar between human cancer cells and mouse cells. But it turns out as you go further and further into flies and worms, the identities of the microRNAs are different. And that's because these animals use very different modes of regulation for their different sort of strategies. So we haven't looked too much into cancer cells, but I think this process is definitely active and it could be exploited for therapeutic benefit. At least you can imagine doing so.
And I just learned that that question was posed by Mehmet Tolga, class of 73. Thank you, Mehmet. Mark Snyder asks, do short half-life microRNAs have something in common in terms of the mRNA that they target?
Yeah, another really interesting question. As far as we can tell, the commonality in targets don't seem to so much fall into sort of gene ontological subsets, at least not in an obvious way that we've looked. But something I didn't really mention as much is that there is a very unique type of microRNA target pairing that characterizes these fast turnover, these short-lived microRNAs. So something I didn't quite say is that normally when microRNAs function to repress mRNAs, they're only using the first part of the sequence of the microRNA to base pair to the target. So this microRNAs are about like 20 something nucleotides long. They're only using about the first 7 or 8 nucleotides usually to target mRNAs. And so that's the regular pathway. But this degradation, microRNA degradation pathway actually involves much more extensive pairing to almost the entirety of the microRNA. And that's actually really quite rare in the transcriptome. So not a lot of those sites exist. And perhaps part of the reason is because these sites tend to cause the microRNA to be degraded. And so this is kind of a special molecular architecture that cells use to tune the accumulation and the sort of lifetimes of these microRNAs. So just to reiterate, basically every microRNA that is fast turnover for which we know the target that's responsible for that fast turnover has this really extensive pairing to that target.
And sort of two related questions that I'll sort of link. Do any of the microRNAs show different or significantly different half-lives in different cell types? This is from Charles Hoffman. And a related question from someone whose name I don't know. Are there examples of the same microRNAs exhibiting differential stability in different cell types? So both questions get at cell type specificity of the degradation.
Yeah. So this is, I think, a really important thing to consider. The answer is, in short, yes, there is definitely cell type specificity for in determining which microRNAs in particular are affected. And what we find is that basically a big determinant of which microRNAs are being degraded in which cell types is actually the expression level of the target, which causes their degradation. So we've found examples where there's kind of a threshold of expression level for that target, below which the microRNA that binds to it in such a way is safe, essentially. It doesn't really get turned over quickly and is stable like usual. But above the threshold, the microRNA then becomes targeted itself for degradation. So I think a large part of the cell type specificity has to do with differences in just gene expression. You can imagine that this is a way in which different cell types and different tissues might carve out different profiles of microRNAs that get expressed.
And I think the last question that I'll pose is from Antonella Catanzaro. I apologize if I messed up your name. What are the effects of overactive microRNA destruction, or and maybe more generally, what would happen if you overproduced microRNAs or got rid of some of them?
Yeah, I think we're just at the beginning of figuring out what the biological consequence is of this type of microRNA degradation. So stay tuned. We're still looking in different organisms, but I'll just say a little bit about the few examples we do know. So there is a particular microRNA, for example, that gets degraded, particularly in certain neurons in mice and in zebrafish as well. So there's a study which looked at this interaction and found that when you basically alter that target in such a way that it no longer degrades the microRNA. So if you get rid of the degradation, this microRNA now overaccumulates, as you're suggesting, and actually causes a behavioral phenotype in fish and in mice. And so these animals are behaving differently, and I forget exactly now what the particular phenotype was, but they were definitely significantly different in behavior. And so we expect that for every conserved interaction like that, that there is some type of there is a phenotypic consequence that has been acted on over evolution to preserve that type of interaction. So we're still very much looking into it, but we expect to find some interesting biology there.
Thank you, Charlie. I want to thank the three student speakers for really stimulating talks, really exciting research, and for participating in this event. I want to thank Dean Mavalava for her remarks at the beginning, and the Alumni Association, Daniel Griffin, Lizzie Army, and everyone else who helped make this event possible. And thank you all for participating. I want to invite everybody who still has time, and if you're interested, to join us in a less formal Zoom setting. The link is in the chat if you would like to join, and I will be there shortly, I think along with Daniel and several others. So thank you again, everybody, for participating, and hopefully we can see some of you on campus in the not too distant future. And I look forward to seeing many of you soon in the Zoom reality. Thanks again, and everybody, please be well.